Hey everyone, and a huge welcome to all of Mr. Chee's students and followers. Get ready, because today we are diving deep into one of the most incredible, most important processes in all of biology, meiosis. You can think of it as nature's way of shuffling the genetic deck, and trust me, it's a game changer. Let's get into it.
So let's start with a question that, hits pretty close to home. Unless you're an identical twin, you and your siblings probably look a little different, right? I mean, you've got genes from the same two parents, but the final combo is totally unique to you. Well, the reason for all that awesome variety is the star of our show today, meiosis.
Okay, so to really get why meiosis is so important, you first have to understand the huge problem it solves. See, in sexual reproduction, you're combining genetic information from two parents. Now, if each parent just handed over all of their chromosomes, the baby's chromosome number would double, and then their kids would have double that. You can see how that would get out of control, like really fast. Meiosis is nature's super elegant solution to this problem.
All right, before we go any further, we gotta nail down a couple of key terms. First up, diploid. Most of the cells in your body, your skin cells, your muscle cells, are diploid. The di part means two. It means they have two complete sets of chromosomes. You got one set from your mom and one set from your dad. It's the full, complete instruction manual for how to build and run you.
And on the flip side, we have haploid cells. If diploid was the full manual, haploid is just half of it. These cells have only one single, complete set of chromosomes. And these are your super specialized sex cells, or gametes. We're talking about sperm and egg cells. The whole entire point of meiosis is to take one of those diploid cells and turn it into these unique haploid cells.
So, inside those diploid cells, the chromosomes come in matching pairs. We call them homologous chromosomes. Now, homologous is just a fancy science word that means they match up. Think about it like a pair of shoes. You get a left shoe from your mom and a right shoe from your dad. They're the same size, the same style, they're both for your feet, but one's a left and one's a right. They're a pair, but they're not identical. That's a homologous pair.
All right, let's get into the action. Meiosis is a two-part story. First up, we have meiosis 1, and its number one job is to separate those homologous pairs. It's time to split up those pairs of shoes. This whole stage is called the reduction division, and that name is a huge clue. This is where we reduce the chromosome number, going from diploid down to haploid.
And lucky for us, there's a simple way to remember the four steps. P-M-A-T, PMAT. Prophase, metaphase, anaphase, telophase. In prophase I, those homologous pairs find each other and do something incredible. They actually trade little pieces of their DNA. Then, in metaphase I, the pairs line up right down the middle of the cell. In anaphase I, those pairs get pulled apart to opposite sides. And finally, in telophase I, the cell splits into two, leaving us with two new haploid cells. Now, that little swap of genetic information we mentioned back in prophase 1, it has a very cool name, crossing over. And this right here is absolutely essential for creating genetic variation. Seriously, it's like taking a page out of one book and a page out of another and swapping them to create two totally new stories. This is how you get brand new, unique combinations of genes on every single chromosome.
But hold on, we are not done yet. We've got our two cells, but if you look closely, each chromosome is still made of two identical halves that are stuck together. We call those sister chromatids. So meiosis 2 is the second big division, and its job is to separate those sisters. And if you studied mitosis, this part is going to look very, very familiar.
So we basically run the PMAT playbook all over again. Prophase 2, metaphase 2, anaphase 2, telophase 2. But this time, it's happening in both of the cells we made during meiosis 1 at the same time. The individual chromosomes line up, the sister chromatids get yanked apart, and the cells divide one last time.
So, after all that separating and shuffling and dividing, what do we actually end up with? Well, what we get is nothing short of a biological masterpiece. The entire reason we do this whole dance is for genetic variation. Let's look at the final score.
4. That's the magic number. From that one single starting cell, meiosis churns out 4 haploid daughter cells. And here's the most important part. Thanks to all that crossing over and shuffling, not one of these 4 cells is genetically identical. They're all unique.
So let's just quickly recap where all this amazing variety comes from. There are two main engines driving it. First, we've got crossing over back in prophase I, where the chromosomes trade genes, and second, we have random arrangement. When those pairs of chromosomes line up in the middle during metaphase I, which side the mom chromosome and dad's chromosome goes on is totally random. It's like flipping a coin for every single pair, creating a staggering number of possible combinations.
Okay, let's break this down because this part can be a little tricky. The big difference between the two stages, it all comes down to what is being separated. In meiosis 1, the homologous pairs split apart. That's the reduction step. But in meiosis 2, it's the sister chromatids that separate, just like in mitosis. Just remember, meiosis 1 cuts the chromosome number in half, meiosis 2 just separates the copies. Now for one of the most common points of confusion, mitosis versus meiosis. Okay, for all my students out there, especially the high flyers, this chart is perfect for your study notes. The biggest takeaway is this: mitosis is all about growth and repair. It makes two identical diploid cells. Meiosis, on the other hand, is only for sexual reproduction, and it makes 4 unique haploid cells. Their purpose and their final result are completely different.
You know, meiosis is this incredibly precise, beautiful dance of chromosomes. It's truly amazing. But, like any really complex process, sometimes things can go wrong. And when they do, the consequences can be really significant.
The most common type of error has a name: non-disjunction. Let's break down that word. Disjunction means separation, so non-disjunction just means a failure to separate. It's when a pair of homologous chromosomes, or maybe a pair of sister chromatids, just don't get pulled apart correctly. What happens then is one new cell ends up with an extra chromosome and another cell is missing one. And this number shows you the real-world result of that. In us humans, if non-disjunction happens with chromosome pair number 21, a sperm or egg cell can end up with an extra copy. If that cell is used in fertilization, the resulting baby will have 47 chromosomes instead of the usual 46. This specific condition is known as Down syndrome, and it's the direct result of a tiny mistake in this very complex process.
So, we've seen how meiosis is this fundamental engine, constantly driving genetic diversity in all sorts of living things. It's a huge piece of the puzzle explaining the incredible variety of life on this planet. It really makes you think, doesn't it? What other biological processes take this genetic blueprint and work with it to shape who we ultimately become? It's a great question to ponder.
Hey, I really hope this deep dive into meiosis helped clear things up for you. If you found this breakdown helpful and you want to crush your next biology exam, you have got to join Mr. Cheese community. Make sure you follow and subscribe on TikTok, Instagram, and YouTube.